Hello, students, and welcome to my final video of the year for Unit 5, Chemistry of Life and Cells. I'm going to miss you all so much. This is it for me at TASM. This unit covers a lot of material, but it doesn't go super in depth. So even though it looks like it's a lot of topics, you don't have to know deeply like you did for DNA, etc. So this unit is us thinking about what is the chemistry of life, how are cells made, and then cells coming together to make organisms. And so let's jump right in, and this unit will really help you get prepared for AP biology if you're taking that course. Like all my videos, this is for free, and it's just meant to help students. A couple of tips for you: stop the video often, ask yourself questions, construct your own explanations, use the materials that I gave you in class to practice. Like, look at the Pogles, look at the gizmos, look at the worksheets we did together, look at the Egg Lab, look at the Agar Lab, right? We did all those activities in class, and I might recycle some of those, and I really feel like they help to explain what we're learning about. So, from the study guide, let's just put it all up here real quick about a little bit about chemistry and then about water. Now, these parts for 1 through 3 won't be on the test. You don't have to know about atoms and the octet rule, but it helps you to understand what's going on with water and with the bonds between hydrogen and oxygen. So go ahead and pause the video. Can you do these items? And then check back in with me. All right, so here's a little bit of chemistry review for you if you did any of this in middle school. Here's the periodic table. Each of these represent an element, right? And the smallest unit of an element is an atom. And so we were talking about hydrogen atoms and oxygen atoms, and then we're also talking about carbon and nitrogen and phosphorus as well. And these atoms are most stable when they have 8 electrons in their outer valence shell. That is their preferred energy state. So atoms on the periodic table, unless they're a noble gas, will try to bond or give up or gain electrons till they're stable in their outer valence shell. So let's look at it right here. Oops. Here would be an example of hydrogen. Hydrogen is a special case because the first level only is stable with 2 electrons. And so it just needs one more electron here. Oxygen has 1, 2, 3, 4, 5, 6 electrons. So it needs 2 more in this outer shell to be at its greatest stability. So this provides an example where oxygen can bond with hydrogen or oxygen could bond with itself to give itself 8 electrons in this outer shell and to give hydrogen 2 in its shell right here. So let's see it. This is what I just told you about, that atoms are at their greatest stability with 8 in their outer shell. That gets a little more complicated as you go down the periodic table, but for our elements of life, this holds well. So here's an example of sodium and chlorine, and so this is going to form salt. Sodium, which is Na right here, will give up an electron, and chlorine will accept it. This gives chlorine 8 in its outer shell, and because sodium lost 1 in that shell, it is also going to have 8 as well. All right, so let's see here. Here you can see hydrogen bonding with itself, and so that can show you how it has 2 in its outer shell. And here you can see oxygen bonding with 2 hydrogens to make H2O or water. And let's look at it for oxygen. 1, 2, 3, 4, 5, 6, 7, 8. And let's look at it for hydrogen. 1, 2. When they share, it counts for both. And so this is called a covalent bond, the sharing of electrons. This is very strong. This is going to be an intramolecular bond, so within the molecule. And that's how water is made. Now, you know that water is really important to life. Water makes life possible. It exists as a solid, liquid, and gas on Earth. Can you describe why water is a polar molecule? Do you remember? If you don't, you might want to go back to that TED Ed video we watched on it. So hopefully you got something along the lines of the oxygen attracts the electrons more often than they are attracted around the hydrogens. So therefore the oxygen is more relatively negative and the hydrogens are relatively positive. This gives water a polar structure in that it has unique charges. The oxygen is negative and the hydrogens are positive. You might say, well, why is that important? And it's not important until we have multiple water molecules together. So let's see what happens here. Here we have, this is just another example of the oxygen having that slight negative charge and the hydrogens having that slight positive charge. And so here is hydrogen bonding. Water molecules will bond to each other, and they will also bond to other polar substances because they have a charge. The positive hydrogen here bonds to this negative oxygen. This positive hydrogen bonds to this negative oxygen. This negative oxygen bonds to this positive hydrogen. These bonds are between molecules, and they're called intermolecular forces. They often break and reform, but they have an important property. They give water cohesion and adhesion. So let's check it out. Covalent bond within the water molecule, hydrogen bond between the two molecules. All right, and so here's another example of it. So here are some of the emergent properties of water, and that means that many water molecules together can do something that one on its own cannot. Just like if you have multiple players on a basketball or football team, they can do plays that you can't do on your own. So can you describe some of these here at the bottom? Take a pause for a second. All right, thank you for trying that. Let's go through them. So cohesion is that water molecules like to stick to each other, and adhesion is that they stick to polar surfaces. This is really important because it enables transpiration in trees or plants. And so what happens here is the water molecules stick to each other, and they also stick to the polar sides of the walls inside the tree trunk or inside the plant trunk. This enables water to be able to go up the tree, and then it goes all the way to the leaves without any energy being added. As water evaporates through the stomata, remember it was stomata with you, that the next water molecule will get dragged up. If this didn't work this way, if plants and trees had to use energy to transport water, they wouldn't be very high. Let's see some other ones, right? Water helps to cool us down when we sweat. When we sweat, the water molecules with the most kinetic energy are going to leave. And when water evaporates, the surface of the liquid that remains behind cools us down. Water takes a long time to heat up. So if you were to heat up a pot of oil and a pot of water, the pot of oil would get hotter more quickly. And this is why it takes a long time to heat up a lake. Like if you go to Canada during the summer and you jump in a lake in June, it's going to be cold. It takes a while for it to absorb lots of energy. This is good for us because it helps us moderate our body temperature. So we drink water and then we're able to maintain our body temperature within a certain range, which is important for our enzymes. Water expands upon freezing. So for this, you got to use the little magnet kits. And for liquid water, we had them breaking all over each other. But in the solid state, they freeze and the hydrogen bonds form a lattice structure. This is going to, if you look at this picture right here, it's going to increase the relative volume here. And since density is mass divided by volume, if volume goes up and mass is the same, the density goes down. So ice or water in its solid state will float. This is really important. And why is it important? Is it allows life to exist in frozen ponds and in the frozen parts of the ocean. If it didn't work this way, if it froze from the bottom up, then as soon as it got cold, every body of water that freezes would freeze the whole way through. And that would cause the death of all those fish. Water also helps to dissolve substances for us. That would be ionic and polar substances. So here's a good example of sodium chloride. Look at how all the negative oxygens are surrounding that positive Na plus, that cation. And in here, all the H's, which are all positive, are surrounding the negative chlorine molecule and helping to break them apart. This is important for us because as we eat salt, right, we need to break apart the sodium and the chlorine. We use sodium with the sodium potassium pump with action potentials, and chlorine also does some roles in our body. And an example of that being messed up would be cystic fibrosis. All right, there's my kid on Jebel Rockdar. So that was water. You don't need to know a lot about the octet rule, but I do feel like learning a bit about water and the idea of emergent properties is really important for me. We then spent some time learning about carbs, lipids, and proteins, and we really skipped nucleic acids because we spent so much time on DNA in that unit. So, what are carbs, lipids, and proteins? We've heard of them, we eat them. How are they assembled? What's dehydration, synthesis, and hydrolysis? What are the functions of each one? How does the structure lead to its function? These are hard questions. The structure leading to its function, but it's a core theme of biochemistry that the structure of a molecule helps to lead to its function. I don't expect you to have all that memorized, but I want you to think through it and to process it. All right, go ahead and pause the video and see what you can do here. All right. Oh, Legoland. Oh, here's why I put Legoland in here, right? We're going to make some polymers. We're going to put a lot of Legos together like this. Oh, look at my kid. Okay, so a monomer would be like one Lego block. A polymer would be many of them put together. In our body, we need to make polymers of proteins. We need to then also break down polymers so that way we can digest them. For example, when you ate a piece of bread or something for breakfast, you're going to break down some of the sugars inside of it so that way your body can do cellular respiration. So that's monomers and polymers. The way we put them together is through a process called dehydration synthesis. And this is pretty easy to remember because synthesis means to make, so we're making a larger molecule, and dehydrating means we lose a water molecule. So let's look at what's going to happen here. This is a sugar, and this is a sugar, two glucoses. We're going to remove this H and this OH, which is going to be water, right? When they combine together, it makes H2O. So we remove the water molecule. And now we're going to bond on that O, on that oxygen. I'm sorry, it's not shown there. And that oxygen will be able to have its 8 electrons if you look at it, because it's going to bond to this carbon and to this carbon. So that'll be 2 bonds. And remember, it had 6 to begin with. So that's 8. So everything is great. Here, we're going to take this sucrose, this table sugar, and with an enzyme, we're going to break it apart. And that's called hydrolysis. And so this one's a little trickier. You need to just remember that the word lysis means to break, and hydro is with water. So to break with water. So we're going to break apart this polymer, this sucrose, by inserting a water molecule. This is done with the help of an enzyme, and that enzyme is called sucrase. And here we have put the OH here and an H here. And now we have glucose and fructose. Both glucose and fructose can enter the cellular respiration pathway and become energy for us. So without sucrase, we couldn't eat table sugar. And that'd be pretty sad, right? You all like sugar. We don't have some enzymes like cellulase, so we can't eat tree bark. So that's hydrolysis. Okay. This is a hard question. Like, what is a carbohydrate? And we know that it's breads and pastas, and it's what you eat before a race. So tomorrow I'm doing a 10K race. Hopefully, I've been a little ill. I'm sorry, G-Block, that I had to leave your class. That's never really happened to me before. And so I'm going to eat some carbohydrates tonight so that I'm ready. So carbohydrates have a fixed ratio of carbons, hydrogens, and oxygens, 1 to 2 to 1, or pretty close to that. And these are going to be quick energy in our body. We're able to break apart sucrose and have glucose and fructose and quickly do cellular respiration to break those down into ATP. Glucose is the preferred energy source for our cells. And so the structure of a carbohydrate, this ring shaped structure, and the amount of carbons, hydrogens, and oxygens enables it to be quick energy for us. Carbohydrates often end in OSE, like glucose and fructose, sucrose, and maltose. And so that's what I just said. They can also be used for structural purposes like tree bark as well, depending on how they're put together. All right, so carbohydrates kind of hard. Lipids has the same elements in it. It also has carbon, hydrogen, and oxygen, but it has relatively many more carbons and hydrogens in the tail. That's going to enable lipids to store more relative energy than carbohydrates. So they are going to store it. So if we don't use up our carbohydrates, right, they're going to turn into fats. And this is what a fat looks like. A fat's going to have a glycerol molecule. Don't need to memorize that, but it's going to have a molecule here and it's going to have 3 fatty acid tails. All right, these fatty acid tails are nonpolar. They don't dissolve in water. And so some examples of fats and oils would be if you put butter or if you put oil on water, it's not going to dissolve in there. It's going to float on top, right? And so these store relatively more energy than carbohydrates. There's some unique lipids such as phospholipids. And so let's take a look at it here. Here is a fat. And so this would be what we call a triglyceride. Saturated fats, which you don't need to know the difference of, have no double bonds. So they have all straight carbons and hydrogens here, and they will be solid at room temperature like butter. Unsaturated fats do have double bonds. I'd have to see one here. Here's a double bond right here. And it causes it to kink, and that makes it liquidy at room temperature. And so that's important. Well, here, this will be a nice example of dehydration synthesis. We'll see the OH and the H get removed. There you go. There goes the water. And this is going to make the fat. So your body's doing this, is making a fat if you didn't run off your carbohydrates. Here's what's unique is that we just did this in cell transport. So you should hopefully feel good about this. But they can make phospholipids, and that's going to have a polar head and a nonpolar tail. And so instead of having 3 fatty acids come off, we remove one of them to make a phospholipid, and it's going to have these carbons and hydrogens tails right here. They're going to be nonpolar. They don't like water. They're going to face the inside. And in where we had that third leg, we're going to instead add some other molecules up here called a phosphate, and even one called choline, and they're going to have a charge. And because they have a charge, you see right here with this O negative, they're going to like water. So they're going to be hydrophilic. This is really important because it forms natural bilayers and it makes membranes. And so without a membrane, like without skin, our stuff would fall off everywhere. Well, that's pretty gross. But without like a container, like for your locker, your stuff would go everywhere. And so without a container or without a membrane, the cell would just dissolve. Steroids also count as lipids. We didn't get to go over them this year, so I will not put this on the test. But things like testosterone, cholesterol, estrogen, because they don't have polarity, they're going to be hydrophobic and they're able to just float into the cell. So lipids and carbohydrates are a good example of how structure leads to function. How the structure of a molecule leads to a function. Even though it had the same carbon, hydrogen, and oxygen, the lipids had them constructed in a different way, and they had relatively more carbons and hydrogens, and so that enabled them to store more energy. All right. How about proteins? This was last year up at Jebel Akhdar at the Anantara. My goodness, what a nice place. All right, so hopefully you're feeling a little good about proteins. Proteins are amino acids put together they're going to have carbon, hydrogen, and oxygen as well. And they'll also have nitrogen, and that's going to be part of the amino acid part. They also will have sulfur. So here is what an amino acid looks like. It's going to have a central carbon. It's going to have what's called a carboxyl group. You don't need to know that, but this carbon and an oxygen and hydrogen and an amino group. And they're going to be able to bond together. And so let's see that happen coming up here. So here's that OH and here's the H. And look at that. We can do a dehydration synthesis reaction to bond two amino acids together. And so the proteins in your body can be thousands or millions of amino acids together in a chain, bonded together. Once they're bonded together, they'll start to twist and fold and make a shape. And we'll see that in a second. But let's watch this dehydration synthesis here. There we go. Now let's see how we break it apart, right? If you ate some chicken or something for lunch, now you're going to use an enzyme to break apart these two amino acids. So then you can use those amino acids to build your own proteins. All right. So one thing I failed to mention there is that amino acids all have the same backbone. Look at how they all have H2N, CH, COOH. But what's different is called the R group. And the R group can be radically different. So look at how different lysine looks from alanine. Very different. Or aspartate from glutamine. And that's going to give each amino acid different properties. Some of them like water, some of them don't like water, etc. And so the protein will start to fold and make shapes. Some of the R groups will actually bond with each other, and they will have ionic bonds where they transfer an electron. Some of them really hate water, so they're going to go towards the interior, the very inside of the protein. I think we did a protein party in here where you got to wear the different molecular, whether it's polar or non-polar, and then move around in a group. All right. You don't need to worry about quaternary structure. That's why I skipped that slide. We ran a little tight on time this year with the UN SDG project, which was awesome, by the way. And I hope you really enjoyed it. All right, so proteins gain their function from the way they fold, and a lot of the time they act as enzymes. They can also act as signaling molecules. For instance, insulin is a protein, and insulin tells your body to bring in glucose. But enzymes is what we studied next. And you all had a lot of fun with this. We did 2 big labs, right? And the big labs we did, we'll come back to that. We'll come back to enzymes. I forgot about DNA. And so here's my cat. I love this cat. It's such a good cat. And if you need a cat, unfortunately, this cat cannot come with us to Taipei, Taiwan. And so we're looking for a home for this kitty cat. And we have food for it, and we have its shots and everything. So let me know. All right, the function and structure of DNA, right? We went over in the last unit, the last time we actually had a test, and that's to store our hereditary information, our genes. And hopefully you remember that those genes are stored in the sequence or order of our base pairs, our A's, T's, G's, and C's. Those are then read by what's called RNA polymerase to make an mRNA transcript, which is then read at the ribosome to make a protein. So the structure of DNA leads to this function, and it holds our hereditary information. And the structure is going to be a phosphate, a sugar, and a base. A phosphate, a sugar, and a base. All right, and they can be linked together, and they can make long strands. And you probably remember that A's bond with T's and G's bond with C's. This is a good example of hydrogen bonding, right? And so we talked about that with water molecules, that the negative oxygen bonds to a positive hydrogen in the water molecule. Well, here, this is hydrogen bonding, even though it's not water molecules, it's bases in DNA. This O in this thymine base, or this oxygen, is going to hydrogen bond with this positive H off of this adenine. This positive H is going to bond with this negative N off of an adenine. And then same thing here. All right, so sorry about that. I went a little bit out of order. Let's get to it now. Enzymes. And so you all did a great job with the enzyme lab, and I hope you really liked it. We did two of them. We did I got a picture of it coming up just to remind you. I'll go back. We did it with liver, chicken liver. Oh, it made the room smell so bad. Apples and potatoes, and then I didn't put a picture of it in there, but we got hydrogen peroxide this year, and we had the yeast spheres that were pretty cool. And you tried that with different substrate concentrations, and that was pretty fun. So I hope you really enjoyed that lab. You did a great job. All right, what enzymes are? Is enzymes are proteins that help to speed up chemical reactions. And so they're specific. They only work on a set reaction. So we have thousands of enzymes per cell. They're really important. These reactions might happen normally anyway, but they wouldn't happen at a rate fast enough in our body to sustain life. So without enzymes, we would be dead. Enzymes, let's look at this graph right here. They're going to help to lower the activation energy needed to rearrange those chemical bonds. So in this example, we need to put a little energy in to start rearranging chemical bonds. And maybe this is going to be H2O2. And we're going to rearrange these chemical bonds to make hydrogen peroxide into water and oxygen. So now we've gone from something that's a 2 in front of there, just so it's balanced. We've gone from something that is toxic for us to something that's not toxic for us. And so that's with the help of an enzyme, and that enzyme is called catalase. Enzymes don't all, this would be an exergonic reaction. This gives off energy. If you remember, you held the test tube and it got hot, right? You could feel the energy going off. Enzymes can also help to build things up to make bigger molecules. For instance, there are enzymes that are going to help to put together carbon dioxide and ribulose bisphosphate and other molecules to make sugars, which we learned about in photosynthesis. All right, let's do this. So nothing works without enzymes. We need them to put together molecules and to break them down. Enzymes are called catalysts. They speed up chemical reactions. Enzymes are specific. They must have the right shape for the job. They're often named after what they do. So, and they often end in ASE. That's a trick for you. So sucrase breaks down sucrose. Proteases break down proteins. Lipases break down lipids. DNA polymerase builds up more DNA. Enzymes aren't used up. They can be used over and over again. So here's the substrate. Here's the active site. This is the enzyme, the purple one here. And the purple one's ready to be used again, just like the key to your house. You can use the same key over and over and over again, but it only works on your house. And that's why we call it the lock and key model. So here is a nice example of it. Start with number one. The enzyme sucrase is going to bind sucrose. It's going to lower the activation energy needed to break those bonds. The substrates will be released with hydrolysis, and then the active site's available for another molecule of the substrate. And so here is all the vocabulary. If you need to review it, the active site is the area that the substrate bonds to. Enzymes are sensitive to changes in pH or temperature. If we change the pH, what happens is we change the shape. And if we change the shape of the protein, it might not work anymore. The substrate won't fit there. Likewise, if we change the temperature, we can also change the shape. It'd be like cooking an egg, and you're never going to get it back. And so here's temperature. Here you can see in this example, here is starch and here's amylase. Amylase is an enzyme that's going to help to break down starch. But here, if you make it really hot, it's not going to work anymore. Look at how the active site has changed, and it will not be able to break down that the substrate anymore. This is a little complicated, graphs with substrate concentration and enzyme concentration reaction rate. But once you get the hang of it, and once you kind of view it through a logic lens, you'll get it. Let's look at what this graph is saying. This graph is saying is as we increase the amount of substrate, let's pretend that the substrate is sucrose or sugar, and the enzyme is sucrase, which is breaking it apart. So as we add more sugar to the system, the rate of reaction goes up. Well, that makes sense. Now we're able to make more glucose and fructose. But eventually it'll level off because we only have so many enzymes. So once we've filled up all the enzymes, if we keep adding sugar, the enzymes are working as fast as they can. And so this is an enzyme reaction rate graph here, or substrate concentration graph. So here's another picture of it. So it's going to level off. And then if you stop adding it to the system, right, it would go back down because eventually we would use up all the substrate. Same thing for adding more enzymes. So let's look at that one here with this pink and green right here. When you add the enzymes, so here the enzyme is the blue part. Once again, let's pretend it's sucrose and sucrase. We add more enzymes, the rate of reaction is going to also increase until we get to the point where the enzymes have used up all the substrate. Then it's going to level out and go down. So here we go. We add more enzymes, and bam, at some point we'll use them all. I went over this with the idea of volleyball setters in class, right? You can have a maximum of 6 volleyball players in the court. So if they're all setting balls over, you could have 6. But if you had more, right, unless they're really good, they can't set 2 balls at once. That, students, is the chemistry of life. Now we're on to cells. Unfortunately, and I'm really sad about it, but we are not going to be able to do endosymbiosis with this quiz. I will go over it just because the story of Lynn Margulis and what she did is really interesting, and we have some cool activities with it. But for this, you just need to know prokaryotes, eukaryotes, plant and animal cells, and cellular organelles. All right, let's do it. So go ahead and pause the video, and can you describe what's going on here? All right, let's just do a quick recap of all the stuff we've been learning. So we started out by talking about atoms. So here would be a hydrogen atom. Those atoms bond together to make molecules in order to have stable valence shells, in order to have 8 in their valence shells, or in the case of hydrogen, in order to have 2. These molecules can come together to make macromolecules, such as carbohydrates, lipids, proteins, nucleic acids. When we put a bunch of macromolecules together, we can make an organelle inside of a cell. For instance, we can make a nucleus that has DNA and it has a phospholipid bilayer around it. If we put a bunch of organelles together, it can make a cell. And a cell is the basic unit of life. Nothing below a cell is alive on its own. If we put a bunch of similar cells together, we can make tissues such as heart tissue or muscle tissue. Those tissues combined together can make an organ like your heart or your brain, and they can work together in an organ system like your respiratory system or your cardiovascular system. So cells, tissues, organs, organ systems, and then the organism is the biggest. So here's like a muscle cell, muscle tissue, organ, the bladder, and then your organ system, your excretory system. All right, why are cells small? So this year, instead of making it pink, I had them be blue. I hope you liked the lab. I hope you found it to be a lot of fun. And we use the same trays right here, right? You put them in, you put the cells in there. And hopefully you have the idea that cells have to be small to maximize their surface area to volume ratio to maximize the rate of diffusion. So the larger the surface area to volume ratio, the quicker oxygen can diffuse in and that carbon dioxide can diffuse out. So here, if this was one big cell that was about 3 by 3 by 3, right? Its surface area is going to be 54, its volume is going to be 27, and its surface area to volume ratio is 2 to 1. This is hard for students. The bigger cell has a smaller surface area to volume ratio than the smaller cell. They are inverse. The smaller cell has a larger surface area to volume ratio. So if we're an oxygen molecule trying to make it to the mitochondria, we got to go all the way into the middle here. And if it takes a long time, the cell could die. Likewise, we need to get waste out. We need to get CO2 and other waste molecules out of the cell. And if it takes a long time to get out of the cell, it could make it too acidic and die as well. So cells have to be small. In fact, cells have lots of adaptations to increase their surface area. For instance, they might have folds like this, like your intestinal cells. Now, you might say, well, then why isn't a cell infinitely small? A cell can't be infinitely small because it still needs to contain proteins and enzymes, and it still needs to be able to do its job, right? Its job might be to send nerve signals, or its job might be to contract and to move your bones. All right. Here are the real reasons that cells are small. Surface area to volume ratio, as I said. Also, diffusion, as we mentioned there, for oxygen and CO2. It needs to be a short distance. And DNA also limits cell size. There's a limit to how quickly DNA can be made into proteins, and cells need a lot of proteins to perform all these functions. And so if the cell is too big, it wouldn't be able to make enough proteins. Here are the size of cells. So bacteria cells or prokaryotic cells are about 1/10 the size of eukaryotic cells, which would be animal or plant cells. They're all really small. Oops, here's a slide from my first year teaching. Looks like the animation got a little jacked up. But just a little graphic organizer here for you. Prokaryote versus eukaryote. Prokaryotic cells are bacteria. They have no nucleus, no organelles. They've been around on Earth for a longer amount of time. Eukaryotic cells evolved from prokaryotic cells. They are animal cells, such as your intestine or your heart cell. There are plant cells that can do photosynthesis. They have a nucleus and they have organelles. So you go ahead and compare and contrast them. And then here are your answers that you can check. All right. I'm going to try and finish strong here, kids. I'm not feeling my best. All right, I'm back. Got some water. So can you compare and contrast plant and animal cells? So both of them have nuclei. They both have a nucleus. They both have endoplasmic reticulums. They both have mitochondria. But the plant cell has a couple things that the animal cell doesn't, like a cell wall to help maintain its shape. And it's going to have chloroplasts that enable it to do photosynthesis. It'll also have a much larger vacuole that can store water. All right. Try this chart. So go ahead and pause the video. Let's see if you got it. Bacteria have a cell wall. They have a cell membrane. They do not have membrane-bound organelles. They don't have a nucleus. They do have DNA. It's just in a region called a nucleoid. Example would be bacteria. E. coli would be an example. Plant cells have a cell wall. They have a cell membrane. They have membrane-bound organelles. They have a nucleus and a cactus. All right, here's your first difference. Bam, animal cells, no cell wall. They do have a cell membrane. Every living cell has a cell membrane. They have membrane-bound organelles. They have a nucleus. Example could be a human cell. All right, the structure of an organelle leads to its function. So I'm going to go a little quick here. I'll link some of my old videos that go over it more. I'm just kind of fading a bit here. We can think of cells having 3 jobs: to deal with energy, to make proteins, and to export them, and to make more cells. And that's a handy way to learn the organelles instead of just memorizing them. So think about them in context. So the cells that deal with capturing and storing energy are mitochondria and a chloroplast. You could even say the cell membrane because it brings in food. Here's a picture of a cell from Khan Academy. So let's go through the items here. So we'll start here with DNA. DNA is inside the nucleus. It's going to have our hereditary material. There's a section of the nucleus called the nucleolus, and the nucleolus is going to help make ribosomes. Attached to the nucleus is the endoplasmic reticulum. The endoplasmic reticulum is a series of membranes that are studded with ribosomes. That would be the rough endoplasmic reticulum. And the rough endoplasmic reticulum is where protein synthesis occurs. The smooth endoplasmic reticulum that doesn't have any ribosomes on it will help to make lipids and deal with detoxification. Once the protein leaves the rough endoplasmic reticulum, it goes to the Golgi apparatus. It looks like these pita pockets right here. And in here, the protein is going to be modified before export from the cell. Let's see some other items. We have mitochondria, which deal with making energy. Mitochondria were once their own bacteria. They were brought into the cell. Mitochondria had their own DNA. They divide independently of the cell. Here's a vacuole. Vacuoles are storage areas where water can be stored and other items. Here's the smooth ER. The peroxisome, you don't need to know, but it deals with breaking down hydrogen peroxide. The plasma membrane is going to separate the cell from the outside world. The lysosome deals with breaking down foods and old dead cell parts. The centriole will deal with cellular division, and we haven't gotten to that yet, but we're going to do a lesson on it. I know I keep saying we're going to do a lot of lessons, and we don't have a lot of time. It's May 16th. I wish school lasted an extra couple weeks. All right. How does the structure of a cell lead to its function? Well, you can have many different cell shapes and types, and they're going to perform certain functions. Here's another picture of the of protein synthesis. We call this the endomembrane system. And this is helpful to know for AP Bio. We never got to this. I'm going to give it to you today, but there's the cellsalive.com. You can play with these. The .com website's now offline, but there's another one that I've linked to Classroom. We're going to go over this more in just a second. All right, you can spend some time practicing right here. All right, see if you can do these parts of the cell. And here we go. All right, practice. Sorry. Now practice. Come on, John. Okay, and here you go. All right, last bit here, cell membranes and transport. I like to think about it like your hallway. Look at this. This is the old high school hallway when I first made this video. You know, it's so much nicer now with all those pictures. I love it. All right, try these questions. We're not going to get to the sodium potassium pump. So number 6, you can cross out. So many things. We just need more time. All right. The cell membrane is going to have phospholipids. They're going to have a polar head and a non-polar tail. The polar head is going to like water and the tail won't. And so these are, their own lipids, but they will naturally form a bilayer or a double layer. And this requires no energy. They'll naturally do this. This is important. It helps to, keep the cell separated from its environment. That membrane is fluid, right? It's going to be constantly moving around. It's going to have channels through it. It's not just like a sheet, like it's fluid. Oh, yeah, we did the bubble lab, and hopefully you saw it there. Things that can easily cross are lipids because they just don't have a charge, and in small nonpolar molecules like carbon dioxide and oxygen. Water can leak through. Things that can't cross would be charged ions like potassium or chlorine, or sugars. All right. And so, well, how can those items get in if they can't cross? They're going to have specific channels. And this brings us to the idea that we need to understand a bit about diffusion. So, diffusion is the movement of particles from high to low concentration. In a liquid and a gas, the particles are moving randomly, random movement. And so over time, that random movement will have them take the shape of the container. It'll have them spread out throughout the room. That's why C block, when you keep spraying perfume and Axe, it eventually spreads out all over the place. Same thing in water, right? It'll spread out throughout the water. So if you put food coloring in water, right, it will spread out eventually. And we did that in class. Some classes I forgot to, but I will do it today. I'm sorry. Okay, so that's the concept of diffusion, or the movement of molecules. So let's talk about a cell and the cell membrane. It's going to keep it separated from the outside. Well, it needs to get certain things in. What does the cell need to get in? It needs to get in food, maybe sugars, bigger sugars. It needs to get in oxygen. It might need to get in signaling proteins. It might need to get in a steroid like a testosterone or a growth hormone or something to tell it to do something. These are all things that need to come in. What does it need to get out? Well, it needs to get out waste. It needs to get out carbon dioxide. It might need to get in or out water. So this happens through diffusion. Sometimes this can happen with no energy required, and that's going to be called passive transport. And sometimes we have to use energy to go against the concentration gradient. So this is a flow chart I got from Bozeman Science. It's really good. So this is cellular transport. There's two main types. There's passive and active, okay? Passive doesn't require any energy. That is diffusion. There's no energy required. Facilitated diffusion, also no energy required, but it uses a helper protein. So the examples we use in class for diffusion would be oxygen and carbon dioxide. And for facilitated diffusion, the one we used in class was glucose. The diffusion of water is so important that we give it its own name. So that way when we say the word osmosis, we know that we're talking about the diffusion of water. And that creates hypertonic, hypotonic, and isotonic solutions. In active transport, we're going against the gradient. We're going from low to high. For instance, taking a bucket of water, lifting it from a river and lifting it from below a dam and putting it above a dam. And we'll talk about that in just a second. Oh, this is from the gizmo. If you got to do the gizmo, it's really cool about the train wreck. So which way will the fats move? Here, they're going to move from high to low, from outside the cell to inside. Sugars are going to do the same thing, high to low. You might be able to see how we can keep a gradient, right? Because we keep using up the sugars inside the cell. All right, occasionally we need to go against the gradient. So look at this. These green ones here, there are fewer on the inside than on the outside. All right, so we're going from low to high. And to do that, to go against the concentration gradient, we need energy in the form of ATP. That ATP is going to cause a shape change on that protein and move sodium and potassium against their gradients. We will try and practice this with the model kit today. And the reason that we do this is we need an excess of sodium outside of our cells for action potentials. And we need an excess of potassium on the inside. And so that's how we send signals throughout our body. Okay, yeah, the egg lab, that was pretty cool, right? You all did a good job with it. Thank you, D and C block, for coming in. G block, I'm sorry I had to leave for it, but we will finish it up next week. And so we dissolve the shell and then we put it in different solutions. So which way will the water flow? So let's look at this, right? 45% water inside the cell, 75% water outside the cell. Water is always going to flow from areas of high concentration to low. So it's going to go from the outside to in. We call this a hypotonic environment relative to the cell. It will make it expand. In this one, we have 90% water on the inside and 63% of the water on the outside. We call this a hypertonic environment relative to the cell. Hypertonic relative to the cell. The cell will shrink. It will lose water. The water will go from 90 to 63. Here, where they're even, 50 and 50, we call that isotonic. So in other words, water is happy, it's even. It will come both in and out at the same rate as it maintains dynamic equilibrium. One way I remember this is hypotonic makes it bloat up like a hippo. Hypertonic makes it shrink like a little kid. And isotonic is the same. So here's hypotonic. And here is hypertonic, so salt water. And here's isotonic. Students, that's all I got for you. Here's some examples of it, right? We use salt water for sore throat. We spray distilled water on vegetables in stores. We have, if you drink too much water, it could hurt you, and water intoxication or hyponatremia. So think about these and think about hypotonic and hypertonic and isotonic environments. Have a good one. Study hard.